But in those days, we could not understand the reason why a normal cell transformed into a neoplastic cell, or why a normal cell starts growing in a neoplastic fashion. But now, in modern era, we can understand why a given cell becomes neoplastic. So suppose here there is a tissue, and obviously this tissue comprises of cells. Okay. Now suppose that this red cell, what you can see, is a neoplastic cell. Now why? What might be the reason for this cell to become a neoplastic cell? See, we have got a multiple reason for a normal cell to be getting converted into a neoplastic cell. One of the reason is DNA damage. See, because it is DNA, and in the DNA we have got genes, and these genes will regulate proliferation and growth and survival of a cell. A cell survival is biologically controlled by its genetic material. So there are genes that initiate proliferation, there are genes that stops proliferation, there are genes that determines how long a cell will survive, and there are genes that will determine when the cell will commit suicide. So very briefly we shall talk about these things. So suppose there is a cell, and in this cell we have got a gene, and the property of this particular gene is that whenever this gene is activated, or whenever this gene is stimulated, it will cause proliferation of the cell. Okay, so whenever this particular gene is stimulated, it will lead to cell proliferation. Cell proliferation means multiplication of the cell, and the name of this gene is proto-oncogenes. Okay, so every cell have got a proto-oncogenes, and whenever this proto-oncogenes get stimulated, it will lead to proliferation of a cell, right? Next, we have got another gene. Okay, and this gene, when this gene will get activated, it will cause inhibition of proto-oncogene, and the name of this particular gene is tumor suppressor gene. Okay, so now until here what you got to understand? Yes, every cell have got two genes. First one is proto-oncogene, and secondly tumor suppressor genes. Activation or stimulation of proto-oncogene will lead to proliferation, and activation of tumor suppressor gene will inhibit proliferation. Why proliferation is inhibited? Because tumor suppressor gene will inhibit proto-oncogene, because of which proliferation will get inhibited, right? Now of course, when a cell will proliferate and when cell will stop proliferation, both this thing depends upon the action of proto-oncogene and tumor suppressor genes. So cells rate of proliferation depend on action of proto-oncogenes and tumor suppressor gene. Now, apart from start and end of proliferation, there is another important thing that you should know, and that is, what is the life of a cell? See, in the cell, there are some genetic programs that determines that how long a cell is going to survive, and these programs that determine the cell should eliminate. Okay, so there are group of genes. Okay, we have got a group of genes. So apart from proto-oncogene and tumor suppressor gene, we have got a third gene, and because of this third gene, what this gene will do, whenever this gene will get activated, it will start producing some kind of product. Okay, we have got a third gene apart from proto-oncogene and tumor suppressor gene, and this third gene, it will be continuously producing some kind of product, and as long as this product is released from this gene, the cell will be surviving. Okay, so survival of the cell depends upon as long as this product is released from the gene, and the name of this particular gene is anti-apoptotic gene. Okay, so the third gene is anti-apoptotic gene. So what this anti-apoptotic gene will do? It will release some kind of product, and as long as this product is released, this cell will be surviving. Okay, next we have got another gene that you can say suicidal gene. Okay, we have got another gene, and this gene is also producing some kind of product. Okay, and when this fourth gene is producing the product, this will cause death of the cell. Okay, so the name of this gene is apoptotic gene. Okay, so we have got anti-apoptotic gene and apoptotic gene. See, apoptosis itself means programmed cell death. Okay, so apoptotic gene will cause suicide of the cell. It will inhibit or it will cause death of the cell. Whereas anti-apoptotic gene, what it will do, it will be releasing some kind of product, and as long as these products are being released, the cell will be surviving. Okay. Now, for a cell to normally function, they must have a functional proto-oncogene and tumor suppressor gene, which will determine the speed of proliferation. And they should also have an appropriate balance of activity of anti-apoptotic gene and apoptotic gene, okay, which will determine the survival of the cell. Now, if we want to convert this normal gene into a neoplastic cell, okay, if you want to convert a normal cell into neoplastic cell, then what should happen? So, for conversion of a normal healthy cell into a neoplastic cell, for happening this, first of all, this proto-oncogenes, it should be overexpressed, and tumor suppressor gene, it should be understimulated. Okay, proto-oncogene should be overstimulated and tumor suppressor gene, it should be understimulated. Such abnormality in a gene is called as mutation. So, to convert a normal cell into a neoplastic cell, there should be mutation in the proto-oncogene in such a manner that it is overstimulated, and there should be mutation in the tumor suppressor gene in such a way that they are understimulated and cannot control the overproliferation. Okay. Plus, there should be mutation in the anti-apoptotic gene in such a manner that anti-apoptotic gene should be overexpressed or overstimulated, and the apoptotic gene should be understimulated. Okay. So, it means that what should happen? First of all, proto-oncogene, there should be a gain of function, and tumor suppressor gene, there should be loss of function. And when this happens, it will lead to overproliferation, isn't it? Because proto-oncogene, it is overstimulated, it will cause overproliferation, and tumor suppressor gene cannot control it. Second thing is mutation. Second mutation that should happen is the anti-apoptotic gene, there should be gain of function, and apoptotic gene, there should be loss of function, which will result in over survival of a cell. Okay, so all these things at multiple level disturbed, a cell may be transformed into a cell which are over proliferated and over survival. Okay, but our biological system has some backup mechanism. See, normally why these genes got mutated? Maybe some radiation induced or some chemical induced, or maybe some inherited defect of any one gene. So, there are so many reasons why mutation can be induced. But remember, every time when there is damage to a DNA, it does not become a permanent damage because it can self-repair. So, we have got a group of gene that produces some product which can repair the damage in other genes, and we call it as a DNA repair gene. Okay. So, now in a normally all these 5 sets of genes should be normal, but to develop a neoplastic transformation, it needs multiple mutation. It needs a series of mutation. Okay, so for becoming a neoplastic gene, all these 5 set of genes should be undergoing mutation. They should not perform their work properly. Okay, so this is how we can convert a normal cell into a neoplastic cell. Okay, so in a molecular era, we can define neoplasia in some different manners. Like in modern era definition, we have got a neoplasm can be defined as a disorder of cell growth that is triggered by a series of acquired mutations affecting a single cell and its progeny. Okay. So suppose here you can see, downwards left side I have made an image where you can see again a red cell, which is a neoplastic cell. Okay, so it is a neoplastic cell and it proliferates. Okay, this neoplastic cell proliferates and it makes many cells. So all the cells which is derived from this specific neoplastic cell are called daughter cells, or more accurately they are called as progeny of the cell. Okay, so to convert a normal cell into a neoplastic cell, there have to be series of genetic damage to all the system, and by acquiring a series of mutation in a cell or its clonal progeny cell until they have tendency to make a abnormal mass of tissue, which will eventually show characteristics such as excessive growth, uncoordinated growth, and there should be persistent growth. Okay, so this was our modern concept of neoplasia, and this is how we can convert a normal cell into a neoplastic cell.